In the deep, silent architecture of the living world, far beneath the threshold of human vision, a war has been raging for more than 3 billion years. It is a silent conflict, fought not with fire or steel, but with information, a ceaseless battle of codes and sequences occurring within the gelatinous, primordial suspension of the single cell. Here in the microscopic wilderness that coats every surface of our planet, from the scalding vents of the ocean floor to the frozen soils of the tundra and the deepest recesses of the human gut, bacteria and viruses engage in a relentless dance of invasion and defense, a biological arms race that has driven the evolution of life since the very dawn of the biosphere. It is a world of unimaginable density and complexity, where the bacteriophage, a virus that hunts bacteria, descends upon its prey like a lunar lander touching down on a hostile planet, its geometric, proteinaceous legs gripping the cell wall with mechanical precision before driving a hollow needle through the membrane to inject its own genetic code, a parasitic command seeking to hijack the machinery of the host. For eons, this has been the fundamental struggle, the invader seeking to rewrite the cellular operating system to produce more copies of itself, and the host seeking to recognize, block, and destroy the foreign code before the infection consumes it. For most of biological history, we believed this struggle was purely instinctual, a chaotic scramble of random mutations and brute force defenses, where survival was merely a game of numbers and chance. Before we go deeper, if this calm pace feels good, consider subscribing so more chill science waits for you when you need it. We looked at the simple prokaryotic cell, the bacterium and the archaeon, and saw primitive entities, devoid of memory, devoid of the sophisticated, adaptive immune systems that grace the vertebrates. We believed that bacteria could not remember their enemies, that every infection was a new event, a fresh catastrophe to be weathered or succumbed to without the benefit of past experience. But we were wrong. Hidden within the twisted, double helical strands of bacterial DNA, buried amidst the chemical letters of life, lay a secret archive, a distinct and peculiar pattern that had been staring scientists in the face for decades, yet remained utterly indecipherable. It was a pattern of repetition and silence, a sequence of genetic code that read the same forwards and backwards, a palindrome followed by a unique jumbled spacer, and then the palindrome again, repeating over and over like a stutter in the text of life. This was the clustered, regularly interspaced short palindromic repeat, a clumsy acronym that would eventually be smoothed into the sharp, crisp sound of CRISPR, a word that now heralds the greatest revolution in the history of biology, and perhaps the history of our species. To understand the magnitude of this revolution, we must first slow our perception and drift back to the humble, sun-drenched origins of the discovery, away from the gleaming high-tech laboratories of modern synthetic biology and into the salt-crusted marshes of Santa Pola on the coast of Spain. It is the early 1990s, and a young doctoral student named Francisco Magica is peering into the biology of Halophrax mediterranee, an archaeon that thrives in environments of extreme salinity, places where the water is so thick with salt that it would desiccate ordinary life in seconds. In these harsh, evaporative ponds, life is stripped to its essentials, forced to adapt or perish. Magica was not looking for a genetic editing tool. He was merely trying to understand how these organisms survived such extreme osmotic pressure. But as he sequenced the DNA of these salt-loving microbes, parsing the chemical strings of adenine, cytosine, guanine, and thymine, he noticed something bizarre, an anomaly that defied the efficient, streamlined logic of bacterial evolution. Prokaryotic genomes are notoriously compact. Unlike human DNA, which is filled with vast stretches of non-coding junk, bacteria usually cannot afford to carry excess baggage. Every nucleotide costs energy to replicate. Yet here in the genome of Halophrax, Magica found a sequence of 30 base pairs that repeated itself multiple times, separated by roughly 36 base pairs of seemingly random, non-repeating DNA. It was a precise rhythmic structure, repeat, spacer, repeat, spacer, repeat. It looked organized. It looked intentional. Nature, in its ruthless efficiency, rarely repeats itself without a purpose. If a sequence is conserved and duplicated, it implies function. It implies that this stutter in the code is vital for the organism's survival. Magica was captivated by this riddle. He found similar repeats in other distinct organisms, separated by millions of years of evolution, suggesting that this structure was not a quirk of the salt marshes, but an ancient, conserved mechanism present across the microbial kingdom. But what was it? Was it a structural scaffold for the DNA molecule? Was it a mechanism for gene regulation, a way to turn biological switches on and off? For years, the mystery of these repeats remained a quiet obsession in the backwaters of microbiology. The scientific community paid little heed to the strange repeating clusters in the DNA of obscure salt microbes. But as the era of genomic sequencing began to accelerate, allowing researchers to read the genetic blueprints of thousands of different bacteria, the pattern emerged again and again, undeniable and ubiquitous. It was found in Yersinia pestis, the bacterium responsible for the Black Death. It was found in Escherichia coli, the workhorse of the laboratory. It was found in the pathogens that plague our hospitals and the beneficial microbes that digest our food. The structure was universal. And then, in a moment of insight that bridges the gap between observation and understanding, the true nature of the spacer sequences, the random bits of DNA sandwiched between the palindromic repeats, was revealed. They were not random at all. When researchers compared these spacers to the growing databases of known genetic sequences, they found a chilling match. The spacers were not bacterial DNA. They were viral DNA. They were snippets of code torn from the genomes of the bacteriophages that hunted the bacteria. The CRISPR locus was not just a structural anomaly, it was a trophy room. It was a biological dossier, a molecular fingerprint file where the bacterium stored the genetic mugshots of the viruses that had attacked its ancestors and failed. This realization upended our understanding of microbial life. It meant that bacteria possessed an adaptive immune system, a form of genetic memory that allowed them to recognize and defend against specific invaders. When a virus attacked and the bacterium survived, it would deploy a complex of enzymes to cut out a piece of the invader's DNA and paste it into its own genome, slotting it between the palindromic repeats like a bookkeeper filing a receipt. But a library of viral fingerprints is useless without a way to use them. The presence of the memory was only half the equation, the mechanism of defense was the other. How did the bacterium use this stored information to protect itself? The answer lay in the Cas genes, CRISPR-associated genes, that were invariably found lurking near the repeat sequences. These genes coded for proteins, molecular machines with specific enzymatic functions. Some of these proteins were scribes, responsible for acquiring new spacers and writing them into the genome. But others, the researchers realized, were executioners. They were nucleases, enzymes capable of cutting DNA. The system worked with elegant, deadly simplicity. The bacterium would transcribe the stored viral spacer into a molecule of RNA, a chemical cousin of DNA that often acts as a messenger. This small piece of RNA, carrying the mirrored sequence of the viral invader, would link up with a Cas protein, forming a search and destroy complex. Imagine this complex as a sentinel patrolling the cytoplasm of the cell. The Cas protein is the weapon, a molecular pair of scissors, but it is blind. It cannot see which DNA to cut. The RNA guide derived from the CRISPR archive is the eyes. It carries the coordinates. As this complex drifts through the cell, the RNA guide constantly checks the DNA it encounters, looking for a match. It is a process of physical interrogation, the RNA strand attempting to bind with every piece of genetic material it bumps into. If it encounters the bacterium's own DNA, the sequences do not match and the complex moves on. But if a virus injects its DNA into the cell, and that virus has been encountered before, the RNA guide finds its target. The sequence matches perfectly. The RNA binds to the viral DNA, zipping together like a fastening mechanism. This binding event triggers the Cas protein. The scissors snap shut. The viral DNA is severed, broken, rendered inert. The infection is halted before it can begin. The bacterium lives. This discovery, solidified in the mid-2000s, was a triumph of basic biology, a profound insight into the microscopic wars that shape our biosphere. But for a handful of visionary scientists, it was something more. It was a revelation of programmability. If nature had evolved a system that used a simple piece of RNA to guide a pair of molecular scissors to a specific sequence of DNA, could that system be hijacked? Could it be engineered? If we could change the RNA guide, replace the viral sequence with a sequence of our own choosing, could we trick the Cas protein into cutting DNA wherever we wanted? Could we turn this ancient bacterial shield into a universal tool for editing the code of life? Enter the laboratories of Jennifer Doudna and Emmanuel Charpentier, two biochemists from different sides of the Atlantic who would eventually converge to unlock the true potential of this system. They focused their attention on a specific protein from the CRISPR system of Streptococcus pyogenes, a protein known as Cas9. Unlike other CRISPR systems that required multiple large protein complexes to function, Cas9 was sleek, a single protein effector that contained all the necessary machinery to cut DNA. It was the perfect candidate for a tool. In a series of elegant experiments, they isolated the components of the Cas9 system in a test tube. They stripped away the cellular noise, reducing the system to its fundamental parts: the Cas9 protein, the CRISPR RNA that held the memory, and a second transactivating RNA that was required to activate the complex. In a moment of bioengineering brilliance, they realized they could fuse the two RNA molecules into a single, continuous strand, a single guide RNA. This was the master key. By synthesizing a guide RNA with a sequence that matched a specific gene, any gene, and mixing it with the Cas9 protein, they created a programmable nanobot. They threw this synthetic complex against a DNA molecule in vitro, and it worked. The Cas9, guided by the artificial RNA, found the precise location on the double helix and made a clean, double-strand break. It was a moment of profound scientific alchemy. They had not just observed nature, they had repurposed it. They had transformed a bacterial immune system into a word processor for the genome. The implications of this transition, from reading DNA to writing it, are staggering. For decades, genetic engineering was a clumsy, laborious process. We were like children trying to fix a watch with a hammer. We could blast genes into cells using gene guns, hoping they would land in a useful place, but we had no control over where they integrated. We could use older editing technologies like zinc finger nucleases or talins, but these were expensive, difficult to design, and bespoke. You had to engineer a new protein for every single target. CRISPR-Cas9 was different. It was democratic. It was cheap. It was easy. To target a new gene, you didn't need to build a new protein. You simply needed to synthesize a new piece of RNA, a process that costs a few dollars and takes a few days. Suddenly, the ability to manipulate the fundamental code of life was accessible not just to the elite genomic centers of the world, but to almost any molecular biology lab with a basic setup. As the news of the programmable Cas9 nucleases spread through the scientific community in 2012 and 2013, it sparked a gold rush of experimentation. Researchers began to apply the tool to everything. They targeted genes in human cells, in mice, in zebras, in plants, in fungi. They showed that they could not only cut DNA, disabling a gene, a process called knockout, but they could also use the cell's own repair machinery to paste in new sequences. When the DNA is cut, the cell panics and tries to repair the break. If a template piece of DNA is provided along with the CRISPR machinery, the cell will often copy that template into the break, effectively rewriting the gene. We moved from merely deleting words in the book of life to correcting spelling errors or writing entirely new sentences. The narrative of CRISPR is often told as a story of speed and conquest, of patents and prizes, but beneath the headlines lies a deeper, more meditative reality about the continuity of life. The tool we use to potentially cure sickle cell anemia, to engineer drought-resistant crops, to perhaps one day resurrect extinct species, is not a human invention in the strictest sense. It is a borrowed technology. It is a gift from the invisible world, a mechanism refined by billions of generations of bacteria fighting for their lives in the muck and the mire. When a scientist in a clean room pipes a solution of Cas9 into a culture of human cells to excise a mutation that causes cystic fibrosis, she is employing the same molecular movements that a Streptococcus bacterium uses to fend off a phage in a festering wound. There is a profound unity in this, a reminder that the language of DNA is universal. The code that writes the bacterium is the same code that writes the human. The scissors that cut the one can cut the other. This universality is what gives CRISPR its power, but it is also what generates the creeping sense of awe and apprehension that accompanies its rise. We have unlocked the library. We have found the pen. The pages are open before us, vast and white and waiting. But the script of life is ancient, complex, and deeply interconnected. To edit one part is to risk altering the whole in ways we cannot fully predict. As we stand on the precipice of this synthetic biology revolution, looking down into the intricate, swirling mechanics of the cell, we must ask ourselves not only what we can do, but what we should do. The silence of the laboratory is heavy with this question. The liquid in the test tube is clear, innocent, yet it holds the potential to reshape the trajectory of evolution itself. We are no longer mere observers of the natural world. We have become active participants, editors of the source code. The rise of the edit is not just a technological milestone, it is a philosophical threshold. We are stepping into a new era where biology is not destiny, but a draft subject to revision. And as we begin to rewrite the draft, we must move with the slowness and respect that such power demands, understanding that every cut we make in the double helix echoes through the complex web of life that sustains us.